Okay. So for today, before we move further in our walk through time, we're going to touch on the basics of evolution and natural selection so that we make sure that we all remember these terms. I am assuming that you've seen at least some of this content before, so hopefully today will be mostly a review or at least partially review. But it's important as we're thinking about, okay, how do we see organisms diversifying and growing past eukaryotes and unicellular organisms and even little small multicellular organisms, that we understand a little bit about how and why that can happen. Tiny class today. Good. This will be hopefully some basics with maybe a few additions checked in. And I am recording, so should be okay on that. All right. So we're trying to figure out how and why early life diversified, like why that could happen. We talked a little bit about things like the changing environment, but we have broader principles at work here. Okay. On the topic of that changing environment, the theory of evolution really says that this is an inevitable consequence of that changing environment. So we see populations adjust genetically to changing environments. And fundamentally, that's really all that this is based on. So we can start with a little thought experiment. So if we have in the lab a male and a female of some new life form that are genetically identical to each other, if we didn't ever change the environment, what do you think we'd see happen? Like probably nothing, right? What would happen if we start to change the environment? Maybe we heat it up, make it colder, throw something else in there. If we change the environment?
They would either evolve or die.
Yeah, right? We might kill them all if we make a really big environmental change and kill off like half of them or most of them. If that doesn't happen at random, we may see evolution. Once it survives, we'll then have altered essentially the genetics of that population. So that's the basis for this. So over our geologic time, we see that life forms are evolving mostly gradually, but we will talk a bit in the future about times when we see explosions of diversity and where we see crashes and extinctions in diversity. So there are sometimes fights between people who study evolution about exactly how gradual it is. I'll talk about that later. So we've talked about how we think this started with a primitive species, maybe a little bit of self-replicating something, maybe RNA. And that started to branch into our diverse species. We can see that we have evidence of things like cyanobacteria pretty early in the geologic record. We talked about how the environment started to fill with oxygen and how we could see that in the rock that we see in deep time. So the mechanism for most of these changes we expect to be evolutionary change through natural selection. And when we think about species, speciation, we are thinking about that change, right? So this is just starting with a probably gradual change over time to this changing environment. But as we see that gradual change, sometimes we're going to have a situation where our population gets split in two, right? Like think about two species getting isolated on opposite sides of a continent, or opposite sides of a volcano that then pushes an island into place. They're really small, so they can't swim around to the other side, something like that. So when we isolate our population, which can happen physically and can happen in a couple other ways, they can then start adapting to their changing environment separately. So they may not do that in identical ways. Those changes that they come up with, those changes that we see in the population, might not be the same changes. So when we think about speciation, we're thinking about splitting two species from each other because of this changing genetics in two populations. We also sometimes see when we're thinking about the fossil record, since we're also adding on the layer of time and we're thinking about gradual change at some point over a long, long period of time, even if you keep that original population together, changes in its environment, changes in where it lives, may over time make it unrecognizable as that single population adapts to that changing environment. So we do have two different terms that we can apply to these different types of situations. Because if you're looking at two organisms in the rock a billion years apart, even if one population just gradually moves forward, you would probably at some point assign it a new species name if it's brown wings or something, right? Yeah. So that gradual difference where we have one species just kind of slowly morph into something looking totally different called anagenesis. When we see a population splitting, so this is where we get our sort of tree of life shape, we call this cladogenesis. When we talk about a clade in evolution or in biology generally, it's kind of like a word for a group. It's slightly non-specific as to whether we're talking about a family, genus, or species, exactly where we're talking about that split, which is why it's a helpful word to use when you're generalizing. So this is cladogenesis, creating two different potentially species if we're talking about speciation, but could be happening at a more fundamental level as well. Should also mention, so when we say a lineage, we're really talking about this line of descent from the parent to the children to the grandchildren and so on. So we're looking at these lineages splitting at a certain point when we're looking at cladogenesis. Okay. So I keep saying the word population because when we're thinking about evolution, we aren't thinking about any one person, any one animal, any one little single-celled organism. We're really thinking about this on a broader level. So we're thinking about this at a whole population level. So when we talk about evolution, we want to think a little bit about the genetics of that population genetics. So here we see 3 different species of birds. We can kind of see them separating out in terms of their genetics, but we also see a little mixture. I really just wanted to have a picture on this slide of populations. Okay. Other words that we want to remember that I'm sure you've seen before, alleles. Haven't seen them before. Genetics is required for a biology major. That's assuming you'll get it in there. So alleles are just variation on the same trait. So 2 different versions, essentially, of a gene, although this class will more often be talking about phenotypes and things we can observe, because when we're looking at these tests, we're looking for things we can observe. So 2 different versions of our, let's say, gene are coded for by a pair of chromosomes. If we list out what's on each of those chromosomes, that's the genotype. If we just list out what we physically see, we're talking about the phenotype, the physical expression of those genes. When we talk about genes and alleles and genotypes, we do refer to whether you have 2 copies of the same allele, in which case we have homozygous genotypes. We talk about whether we have a mix. Then we call this heterozygosity, so 2 different ones. And we think about genes as being dominant, in which case when we have that gene in the individual present in that genotype, we expect that gene to be expressed in the phenotype, so you expect to see it. When we have a recessive gene, the opposite will only be expressed if you don't have a copy of the dominant gene. A lot more caveats to this, but that's our basics that we're starting with for today. So when we're looking at the population level, we can do some calculations with it. And just for my information, have you guys seen these equations before? Okay, so we'll go through them. They're not too bad. But this is looking at population genetics essentially, and the distribution of the dominant and recessive traits. So Hardy-Weinberg equilibrium. Hardy and Weinberg are two different guys who basically came up with a math for this independently. So they both get credit, which is nice. So they both mathematically modeled what's going on in a population from a kind of probability level. So we have two pieces to this. So Hardy-Weinberg equilibrium tells you what you would expect to see if your population is unchanging. So if their genetic distribution across that population is just remaining the same year after year after year. I'm gonna grab something. Be right back. Write down my math because I can't do math while standing and sitting. And I want to tell you this, right? Okay, so here we are looking at our two equations for Hardy-Weinberg equilibrium for what we expect to see. So we're looking at our alleles, P and Q. So we're looking at our dominant allele and our recessive allele. So P, dominant, Q, recessive. Okay, so the first piece of this, if we express basically the percentage of times we see each of these alleles, the probability of these alleles, they're going to add up to 1 if you're just looking at them separately. So when we think about genotypes, we're separating them out from individual genotypes. So we're just saying, like, how many genes in the entire population do you have for brown hair color, plus how many genes do you have across the whole population for blonde hair color. You express that as a decimal, you're going to add up to 1 because those are only 2 options. So that's the first piece. And then the other piece states, so we have another thing that adds up to 1, which is really just if you add up the proportion of your homozygous dominant genotypes, and I'll jump to the end, your homozygous recessive genotypes, plus your heterozygous genotypes, you're also going to add up to 1. Okay? So this is just telling you how things add up in a whole population. So if we look at a group of organisms and we can look at their phenotypes and determine their genotypes, we can figure out when we look at the first generation of the population and then the second generation of the population and on further if we wanted, we can see whether these ratios essentially are staying the same. Okay. So we are going to do some examples. So take a couple notes. So we're going to think about some snails. These snails are either going to be brown or yellow. So we're going to consider our brown allele to be dominant. It's going to have complete self-dominance. So we're not going to go into all the nitty-gritty of how genetics actually works and all the variation that's possible in snails. So totally dominant, brown, totally recessive, yellow allele. So if we start with a population of 1,000 snails, these are parent population, our first generation, we're gonna see in this example that we have 840 of our brown snails. This dominant trait, more commonly seen. We're gonna have 160 yellow snails. So using those equations, we want to figure out all our little variables. So we want to figure out the allele frequencies, the frequencies of the individuals for the genotype, and the number of individuals for the genotype. So I'll flip back to those equations. We're just trying to, actually, we want these numbers. So we're gonna look for P and Q from that first equation that adds up to 1. And we also know that P squared, our homozygous dominant, plus 2PQ, which is our heterozygous individuals, and Q squared, which are our homozygous recessive individuals, is also going to add up to 1. So let's go ahead and pause and do that. So to do this, we're going to jump back and forth between these two equations. So we are going to start with this bottom one. Okay. And which of these variables do you think we're going to look at first? Which of these three things can we actually determine from the information we have? We are going to just figure out both of them. We're going to start with Q. Why does anybody want to tell me why we're starting with Q? So Q is our recessive. Okay, so when we're looking at just the phenotype, brown versus yellow, if we look at the brown one, we know that that's a mix of the homozygous dominant individuals and the heterozygous individuals. So we can't tell apart our P squared from our 2PQ at this point, but we can pretty easily isolate Q squared because that's just the proportion of individuals that are homozygous recessive. We know that the recessive trait is only expressed when we have two genes for it, two alleles for it. Okay. So we're going to start with yellow. So we're going to take our number of individuals that are yellow, which is 160. And we're going to divide that by our total population, because we just want to know essentially our percent. So we had 1,000 to make our numbers easier. All right. So we get that our frequency here of these homozygous recessive individuals is 0.16. And 0.16 is Q squared. Okay. So for this equation now, we could put 0.16 into that Q squared spot. So what do you think I'm going to do next then? How can we start to tease out the parts of the Q? So we're going to take ourselves and jump to that first equation. I'm going to do not disturb. All right. So we have Q squared. And we really can treat it now to get to that first equation as if it's actually a squared number. So we're just going to take the square root of 0.16 to get us Q. Our root of Q squared is Q, which is 0.4. That's 4 times 4. Okay. So now we can plug that in up here. Okay. So now we know P plus 0.4 equals 1. So that leaves us with, perfect, yeah, 0.6 for P. Okay. So now we have our values for Q squared, 0.16, for Q, 0.4, and for P, we have 0.6. Okay. So then we can move back down to that original equation. Okay. So to get our value for P squared, we are just going to square that 0.6. So then we get 0.36. So we have 0.36. We know Q squared is 0.16. And we know we got to add up to 1. So we're going to get 48 left if we do the math out. Or 0.48. Questions about how we did this?
So why did you just say that 0.16 is Q squared? And like, how do you know that that's not just Q right away?
Yeah, so Q squared is by definition, the proportion of homozygous recessive individuals.
Okay,
So we have to use the second equation because this is about those genotypes. So to get that, we just picked our individuals. So this has to do with the individuals, not counting up alleles. So we have 160 individuals that are homozygous recessive and 1,000 in our total population. So that's how we get that frequency there. Does that make sense?
yeah,
And that's basically the reason they state it out that more complicated way rather than just saying homozygous dominant plus heterozygous plus homozygous recessive equals 1. By creating this relationship mathematically, it allows you to jump from those like more observable populations to the total allele frequency and back.
okay,
Does that make sense?
yeah.
Yeah. So that's why it's important to use specifically those variables in that way for that. Any other questions? Okay. Okay. So we're going to do one more step because we're trying to use this to figure out whether we have overall genetic change in our population. Since that's how we're going to get towards evolution, selection, speciation. So we got to do one more generation. So here we're going to do the exact same thing, but with the second generation of snails. So now we're going to be down to a total of 800 snails with the word year for snails this year. Now we have 672 that are brown and 128 that are yellow. So we're essentially going to do that same math and figure out whether the frequency of our alleles has changed. So as we're going through this, somebody remind me what my first step is going to be. We're solving for Q first. So specifically, we're doing that by getting the Q squared number from our homozygous recessive individuals. Okay, so we're dividing 128 by our new total, which is 800. All right, so did that math? What do I get?
160.
Okay, 0.16. Okay. So I didn't mean to torture you. At this point, we got all the rest of those numbers in our original problem just by finding out that Q was 0.16. So actually at this point, we can already stop because we know all the other numbers are now going to stay the same because we're not looking at numbers as individuals, we're looking at like proportions. Okay. So because we have this same frequency here for our Q squared, it means again, we're going to have 0.4 as Q, which means again, P will be 0.6. So I have to add up 1, which means this whole bottom equation will again be this. So extrapolating, because this is the principle of Hardy-Weinberg equilibrium, would you say that this population is in Hardy-Weinberg equilibrium? Yeah, nothing's changed, essentially. We got a smaller number of snails, but we can see that there hasn't really been any change in their genetics. All right. So to end up with Hardy-Weinberg equilibrium and to end up with these equations working over and over again to show no genetic change, we need to make some assumptions. Okay. So the first of those assumptions, the ones I have in bullet points, are going to be kind of set up about their genetics, how the genetics of the organisms are working. So we do need to have organisms that are diploid. So we need to have organisms with 2 sets of chromosomes. So we have 2 alleles. We have something with 4 copies of its genome. The math would get trickier. We need different math. We need them to reproduce by sexual reproduction. If they're reproducing asexually, then that also is going to change how we calculate out these populations. And to make things simple, we need our generations to be non-overlapping. So we need to be able to say, okay, like this is the parent generation, this is the daughter generation. Because if we don't have that and they're mixing with each other, then we don't really know which population to be extracting our P's from. Okay. So those are kind of the setup pieces for the math. And the other pieces have to do with the biology. Okay. So for our population to stay in equilibrium and see no change overall in the genetic distribution, we're going to need to see random mating. So we're going to have to see that all of the individuals in this population have an equal chance of contributing to the pool. We're also going to need a really big population, technically infinite. We don't need any new genotypes or phenotypes arising. We wouldn't know where to put them in the math. So this means that we have no mutations going on here. We also have no migration, gene flow, admixture, which basically means we have no genetic diversity coming in from other populations. So we have like a closed loop here. And we have no natural selection. So we have nothing like picking and choosing which of these genotypes are better for the environment. Okay. So we're going to go through basically each of these factors and how it would affect what we see in the population, how it would affect this genetic distribution, kind of why it matters. Right. So for the first, we're going to think about our population size. Okay. So we need a really large population because otherwise we may see something referred to as genetic drift. And you can think of this as like kind of a statistical thing and just random chance, essentially. If you have a small population, just by random chance, you may see its genetics start to change. So 2 places that we can really observe this happening are through the founder effect and through something called bottlenecks, genetic bottlenecks. So we're going to look at what each of those things mean. Okay. So the first, the founder effect, states or shows us, we can observe this, that if we have a big population And we end up taking just a small little chunk of that population, moving it somewhere else and isolating it. We are going to see that it matters. Shoot, did I close Zoom? We are going to see that it matters which ones he picked, essentially. Okay, so this original population had a population frequency for its alleles. So we had half of them with this red allele, we had half of them with this green allele. So we had an even distribution here. But when we took out just a small chunk here and split it off, we start out with uneven phenotypes. So we grabbed a lot more greens than we grabbed reds. So at this point, we will ultimately end up seeing an effect of this in our final population, depending on what genetics create these specific phenotypes. Okay. So basically, we can start to see, for example, if this red one is quite dominant and more effective, we may see it completely take over here. So this founding population makes a difference. So an example that we have seen in the wild is with some lizards. I think it's the Anatoly lizards. So basically, we've seen some lizards where we had a bunch of different alleles, of different possibilities for the color of our lizards, our initial population. We had some of them migrate to a new island to get kind of isolated there. And we only had yellow ones and green ones. And so our new population, when we eventually look on that island later, we're not going to see those orange lizards anymore. We'll just see yellow and green in some proportion. So that's the founder effect. So this is basically a way we can take out some genetic diversity. Next one, we have our bottlenecks. So bottleneck is when we have a decently large population. We don't take a chunk out, but we have a big dying off so that for a little while we get a really small population. Ultimately, this is going to have a similar result to the founder effect, even after that population recovers, because we've gone through this phase where we don't have as much genetic diversity. And so we are not going to see as much genetic diversity, even once those numbers go back up. So this is, again, having to do with what group you pull out of this original pool. It's just the reason for the pulling out is different. Now, instead of moving a group separate to a new island, we kill the rest of them all. So that's what happens with population bottlenecks. So we could again see changes to the frequency distributions over time. A real life example of one place this has happened is with cheetahs. Cheetahs kind of sad and anxious sometimes. And you'll see articles online asking if cheetahs are all clones of each other. It's not exactly true. They're not exact clones of each other, but they have gone through two really major population bottlenecks. So the first one was when their range expanded. So when they started to spread into Asia and Europe, their populations kind of got spread apart in a way that they weren't able to change their, or they weren't able to have gene flow across the whole population of cheetah. So when the African group was really the only one that survived, it didn't have those genes from the populations that died off. So that was its first bottleneck. And then the second bottleneck is during the last ice age, tons and tons of them died and ended up with a really, really small surviving cheetah population. So even as those cheetahs recovered as the world warmed back up, they lost a lot of genetic diversity in this process. So in 1983, there was a paper that tested a bunch of enzymes in the blood of the cheetah. And in like a normal cat, they found 20 to 50% variation in those enzymes between the individuals. And in cheetahs, I found like basically none. So I pulled out a table from, I think this is a different paper, looking at basically genetic diversity in different species. So this Acinonyx is how I'm going to say that, is the cheetah. I suddenly realized I've never said that word out loud. And we can see cats here. We can see they were also comparing them to humans, Drosophila, all sorts of things. So you can see that as we look towards our genetic diversity, we're going to pull just to the end here. You can see that they had much no heterozygotes. They got a literal 0 figure here for the loci that they were looking at, whereas our other species did have a fair amount of genetic diversity at those same places. So this is part of what they used as evidence for finding those bottlenecks in the past. Okay. So the next piece of those assumptions that we're going to think about, we'll think about two together. We'll think about the random mating part and the natural selection part. These are kind of related. So random mating and assumptions, that's the one where all of our individuals are contributing equally to the gene pool of the next generation. And if we have non-random mating, we may see that our population is changing. So we may see sort of different types of mating preferences. So a mating preference is like literally any preference of the organisms reproducing. So we can have assortative mating, which is when we have individuals choosing other individuals with a similar genotype or phenotype. So they're going same thing. We also see sometimes disassortative mating. So we can have individuals with preferences for genotypes or phenotypes that are different from each other. Both of these exist biologically in different situations and different species, but both of them would be non-random. This isn't just throw them all in a pot and mix them around. So here we can see an example with some mockingbirds. So we can see this mockingbird 1. We have some selection going on. This sad mockingbird was not selected, so has no offspring, but it doesn't leave any genes in the next generation, even if it lives a long time, because of this non-random mating. So it's not that it's dying off in the same way as during natural selection when we kill off a bunch of the cheetahs because they froze to death or something. This is just a fact that is a result of this non-random mating. This mockingbird 2 survives, finds a mate, and it does leave plenty of genes in the next generation. So its genes are then going to be more prevalent in the next population. So an example of this that we have studied is something called the long-tailed widowbird. And I think this experiment is the funniest experiment. So this is an example of sexual selection that they've observed. The long-tailed widowbird has an absurdly long tail to the point that it's actually kind of difficult for it to like carry around and fly. However, these long tails are sexually selected for by females. This is specifically in the male bird, and we watched them do it. So I love biology experiments because what they did, we had our normal widowbirds, and they started either cutting off their tails or pasting on extra tail after trapping these birds. So here we can see the artificially lengthened tails where they just, I'm assuming, I don't know, super glued an extra tail to one of these male widowbirds. Here we can see the artificially shortened group. Got a shorter tail because we cut off part of it. And to check whether we were changing their reproductive success just by stealing them from the wild and doing weird little surgery type things on them, we had a control group where their tails got cut off and then just put back on. So they stayed the normal length. but we could check whether it had to do with the interference of the researchers.
So what we have on the y-axis there is the average number of nests per male. So
It has to do with their territory control and also their contribution to the genetic pool through having different offspring, like numbers of offspring kind of goes with numbers of nests.
so we can see our level for the normal unmanipulated group was a little less than 1 here. We saw the same level for our control group
Where we took off their tails and put them back. So this tells us that the results of the rest of the experiment were not due to the fact that we yoinked them out and messed with them.
So then we can look at the artificially lengthened tails where they pasted on extra and the artificially shortened tails where they just cut some off. We can see a huge difference in how successful these birds were. So the female birds really, really like
The birds with
super extra ridiculously long tails. We saw this is like almost double
the number of nests.
for these experimental super long tail birds. And the poor birds that had half their tail chopped off did really, really poorly compared to even our control groups. So we can see here in this example that we had non-random mating going on. We had sexual selection for these extra long tails, and we could see the birds even preferred if you made this trait super, super extreme. And so this is used as an example of how we can see some of these extreme traits coming into existence, being selected for. And when we talk in the future about like sexual dimorphism, this also contributes. So the next piece we're talking about in terms of what survives and what doesn't is natural selection. So natural selection, this is how, this is how we might have something like a bottleneck happening. So this is going to be non-random contribution to the next generation. And in this case, we can see that our issue is that we have different survival rates, essentially, of our organisms. So in here we can see different colors of beetles. The different colors of beetles are eaten by birds more easily. And so we see a change across generations because if you're eaten, you're not going to have new baby beetles. So the classic experiment for this, or the classic observation, is about the peppered moth, Bison betullaria. So we looked at this around the time of the Industrial Revolution, when all of a sudden cities were filled with tons of smog, and like physically the environment got darker as it was collecting on trees and buildings all over the place. So there are two forms of this moth. There's a dark form and there's a light form. They have the same situation as with those beetles in the cartoon, which is that if they stick out, if you can see these moths, they're more likely to get eaten by their predators, the birds. Okay. So before we had lots of smog in the environment, we have a nice sort of clean kind of forest. You can see here that the light moth is not very visible. It really looks like this lichen covered rock here. And that dark one is really, really super visible. Okay. But if we were to move to a different kind of tree, or if we have smog suddenly settling ash all over the place, we're going to have the opposite phenotype start to stick out. So we saw that all of a sudden the light colored moths are getting eaten more often because they stuck out more. So this has to do with predation. So the predators selecting out different phenotypes, different genotypes, because different phenotypes are produced by different genotypes. So from a population that starts in an environment where our light colored moth is the one that blends in, you'd expect to see that light colored moth predominate. You'd expect to see it more common in the population because that's the one that's surviving. But when we move to a darker environment, we're going to see a change in the distribution of those alleles, the difference in the frequency because we're going to have this dark colored moth surviving longer and therefore reproducing more. So that's natural selection. Doesn't all happen by predation. So we can actually separate different types of natural selection based on what they end up doing to the population. So what they end up kind of doing to the range of phenotypes that we see. So we're going to start by thinking about directional selection. So we're going to start out by thinking about our phenotypes, so our expressed trait, as just a nice bell curve. We see the bell curve or the normal distribution as a distribution that appears over and over again throughout biology, throughout the world. It's kind of the basis for most statistics, is just assuming that things kind of tail off around the average in a specific predictable way. So we're starting with our phenotypic range, just a nice little bell curve. So in directional selection, what happens is that we just have a shift. So we're still going to have a distribution of traits, but the average is going to move in one direction or the other. Okay, so we're going to see a change in the phenotypic range towards one trait or the opposite trait. So an example of this is flowering time of plants. So we're going to start out here with our flowering time of our plants in 1853. We can see them flowering here kind of later in the year. And now, as the environment is warming, our climate is warming, we're starting to see directional selection for much earlier flowering time. Second type of natural selection that we see is stabilizing selection. So we're again starting out with a bell curve. In stabilizing selection, we're going to see that we really start to emphasize essentially the middle of this distribution. So in stabilizing selection, what we're seeing is that basically being weird is bad, okay? So for stabilizing selection, being at this end of the tail distribution, so the tail of the distribution is where it starts to come towards the x-axis, starts to taper down. Either on the high end or the low end in stabilizing selection is bad. So we start out here, low fitness. So fitness, when we talk about the fitness of an organism, that means how likely it is to survive to reproduction and then reproduce and contribute to the gene pool. So if you have the individuals over here and the individuals over there not being very successful, you end up with everybody in the middle. And so you start to see the middle peaking and becoming more common in the population. So a place that we see stabilizing selection can be on things like birth weight. Either being super huge or super small leads to health complications of different types. So we see high mortality rates on the extremes. So you have a 1 or 2 pound baby, that is dangerous for the baby. You have a 10 or 11 pound baby, kind of gets more difficult to be born. So you start to see stabilizing selection on both ends, although with modern medicine, we're kind of removing the stabilizing selection. So these could become more common again with modern medical techniques. But if you have either side dying off, you're going to naturally kind of pick for these middle values. Finally, we have disruptive selection. So this is probably the least commonly observed one, I would say, if we went out and counted around the world, but it is really important when we're thinking about speciation. So we're starting out again with a bell curve. In this case, we're going to have high fitness on both ends, so both of these tails of the distribution, but we end up with low fitness for whatever environmental reason in this central part of our phenotypic range. So this causes the central part of the phenotypic range to kind of die off. So we end up with this kind of bimodal distribution. So we end up with two separate little bell curves now that are more centered around the ends of the previous one. So an example of this is with seed cracking birds. So here we started with a big population of birds that ate different types of seeds. And then we started to see a change in the seeds. So we have some hard seeds, and we have some soft seeds, and we have birds with really long beaks and birds with really short beaks. So in this original population, we had an even kind of distribution. But with these beak lengths, the birds with really long beaks, or longer beaks, I should say, bigger beaks, were really good at eating the hard seeds, able to crack them with their stronger beaks. And so they did pretty well, got lots of food. These birds with the little short beaks were really good at getting the soft little seeds. So they also did quite well. But the birds in the middle were just like worse at both. They kind of could do both, but not very well. Their beaks weren't specialized for either. So they kind of died off. And then we end up with two distributions on either end, one with beaks that are good at hard cracking, the other that was specialized for small little soft beaks. So that would be disruptive selection. The final bit is that we need to have no new genes appearing in the population, which means that if we have Hardy-Weinberg equilibrium, we're not going to see mutation, at least not mutation that succeeds, that produces a new phenotype. And we're not going to see gene flow from other populations, because that basically stops our little closed experiment. Right? So mutations introduce new traits by seeing differences in the DNA. So we have a mutation in a gene. Potentially that mutant copy creates a new allele. Now we have a new option. This can occur through a number of different ways. You can insert parts of genes, you can delete parts of genes, you can substitute parts of genes. You can have all sorts of rearrangements. Okay? So you end up at the population level, ending up with a new genotype if you have a mutation that gets expressed. Okay? So this would not be Hardy-Weinberg equilibrium, because then we have to figure out what happens to that new genotype. Now we have to account for that in our equation. The other bit is gene flow. So if you have a population A over here of bluebirds and a population B on the other side of this mountain of redbirds, they might each be in Hardy-Weinberg equilibrium for a while. But if you then have redbirds start moving across, bluebirds start moving across to the opposite side, you get population admixture, you get mixing, you get migration from one place to another. You're going to be introducing new genes, or at least you're going to be affecting the proportion of genes in that population. So you would expect those gene frequencies to change over time. Cool. All right. So when we break down our ideas about evolution, we can think about different components. Right? So evolution, we're saying it's just that our species is undergoing genetic change over time. So we're going to see changes in the population level genetics. We're going to see changes in what's succeeding and what's surviving, and eventually that'll cause, we'll start to observe that that gene may look really different over time as we select those, and then maybe have mutations that then do survive. Gradualism says that it takes a long time to produce these changes. You need a mutation to arise. You need that mutation to spread throughout the population. You need it to not be harmful. You need it to be beneficial. So we're going to observe this over a long period of time. Exactly how long will partially depend on how long your organism is, how fast it reproduces. So a good place to kind of look for evolution is it like short-lived bacteria and stuff. When we're thinking about evolution, we are assuming, or we are looking at a shared ancestor. So we're tracing lineages. We're starting with that original population and looking at how it changes and how it maybe splits off from other populations, creating speciation. We have natural selection, selecting for different genotypes phenotypes. So we do have other processes that can cause evolutionary change, so change in the genetics of that population over time, like our genetic drift example. Okay, so to look for evidence of this in our fossil record, we're going to look for actual evidence of evolutionary change over time. We'll talk about that as we talk about different species. So all of these we're looking at examples to dive into. We would want to see actual cases of speciation in the fossil record. So we do want to see an ancestral population and an ancestral species giving rise to new species. And we would want to see this in the wild today in order to corroborate our evolutionary predictions. We're going to find examples of species that have common ancestors. So we're going to see these species linked back with common ancestry, where we can maybe trace their genetics, things like that. We're going to see species that have lots of variation in their traits. We're going to see some cases of imperfect adaptation. So this is important if we're thinking that evolution is arising from mutations that get selected for, rather than like designing the perfect thing. You want to see some adaptations in animals that kind of suck a little bit, or there's like an easier way to do that thing, because we're constrained by the biology and constrained by the mechanisms of evolution. So actually seeing something kind of bad helps us when we're looking for evidence of evolution. And we want to see natural selection acting in the wild. So we want to be able to deliberately see certain things surviving and others not. When we're looking at our fossil record, then what we're going to be looking at or looking for is a good selection of sediment and therefore different geologic time periods, right? So to track change over time, we have to actually be able to see multiple time points. Right? Cool. So we have some time points. I'm not going to talk through that. We're limited on time. As we go through, we'll be looking at the common ancestors we see and how we see those splits and how we think about common ancestry, how we figure that out. So we'll be looking at the phylogenetics of these different groups of organisms that we'll be looking at as we walk through time. So phylogeny has to do with tracing these lines of descent through evolution. Phylogeny produces phylogenetic trees where we can trace how and when these groups split off from each other, and we can put on specific traits and see kind of when they appeared by seeing which species that remain have that trait. And so we're going to see nested patterns. And an example of where we can see some selection, kind of thinking in normal everyday life today. Large fish, you know, have many eggs. They have large eggs, large larvae, and low larval mortality. Small fish don't have very many eggs. They're small larvae, high mortality, right? So when we start out, we see fewer small fish than large fish. And the reason that we have fishing regulations to kind of leave large fish in place is so that we have the, other way. The reason you have to leave the small fish is so that they can reproduce and get bigger. We'll talk about this later. This example is not that important and we're at time. Okay. So we will be talking about these different principles as we walk through the organisms that they act on. But this is the basics. And I will speak further about what I meant by that fishing example.